Hey everyone, this is Nick VanAcker. I'm the Science on a Sphere coordinator for the MSU Museum, and I'm excited to bring you guys another Science on a Sphere virtual show. If you haven't tuned into these shows before, we do them every Wednesday at 3 p.m. about all sorts of different topics, and today's topic is going to be about microbes and microorganisms. So we're going to be talking about what microscopic life is, what isn't alive microscopically. We'll be talking about different microorganisms that live on the Earth and in our oceans, and then we'll also be taking a microscope safari at the end of the program. So that should be pretty fun. And giving you guys instructions to do the microscope safari on your own. But if you haven't tuned into one of these before, you might not know, you can actually follow along with these programs at home. So we'll be using an app during the program. I'll be pulling things up on screen, but you can also use the app at home to follow along. And that app is called the SOS Explorer, the Science on a Sphere Explorer. So if you use your Apple or Android device, you can open up the App Store and then download the SOS Explorer app to follow along at home with us. So when we open up the SOS Explorer app, it's going to look something like this. It's got all sorts of different buttons and things you see that you can touch. The first thing we're going to look at is the little green arrows up on the top left-hand corner of the screen. So we can tap that, and you'll see that opens up a menu with all sorts of these different data sets that we can tap and pull up. About all sorts of different things. We can look at things from space, things from the ocean, all sorts of different cool stuff. There's also a category drop-down menu up on the top left. You can see that this will help us sort the data sets if we're looking for something in particular. We'll use that in a little bit. And there are also buttons down on the bottom right-hand corner of the screen. The 2D, 3D button is going to change the way that our sphere looks. So you can see right now it's a flat map we can look at of the world, or we can change it back into a sphere and rotate that. And the final button is going to be the overlays button right next to it. So if we tap that, we can pull things up like country borders, for instance. We can also look at country labels, or we could pull up grid lines for things like latitude and longitude. So I encourage you to mess with those throughout the program, add things if you're interested, and just kind of overlay information on top of the data we're already looking at. So before we get started talking about microscopic life, we're going to have to talk about some stuff that's microscopic but isn't alive. And when I say that, I'm talking about viruses. Viruses have been on people's minds recently, and luckily there's a lot of great resources about them, a lot of great information about them. So we'll only be touching on them today. We won't be really going in depth, but there's lots of other great resources out there all about viruses. But the main thing we'll be talking about, main thing we'll be covering is the fact that even though they're microscopic, they aren't actually alive. And we have a data set we can look at to talk about that. So if we tap the little green arrows, then we can go to the people category, and then we can go down to flu virus model H1N1 2009. And so this data set is showing exactly what the title says. We're going to be looking at an influenza virus, which we're probably all familiar with. Some of us have probably had the flu before. And some of us might have even had this particular flu. H1N1 was a strain of flu that went around in 2009, still continues to go around every year. But you guys can see all sorts of different things going on the globe here. So anytime you see an orange line going from one location to another, that represents one transmission of the flu. So one person with the flu was sick, gave it to another person, that person got sick, gave it to another person. Each of those transmissions from person to person is represented by an orange line. Sometimes there's so many transmissions, so many orange lines that you can't actually see the lines anymore, and it just becomes an orange blob. And you can see the little bar down in the bottom left-hand corner of the screen represents how many people are getting infected with the virus. The lighter colors, represent almost 0 cases, whereas the darker red colors represent up to 10,000 people getting infected with the virus. But the crazy thing about viruses, you see something like this and you might think, oh man, this virus is really mean, it's really malicious, it wants to infect everybody. But viruses aren't actually alive. They don't really have an end goal in mind because they don't have a brain. Viruses are basically just kind of like photocopiers. They aren't alive, but they do still have some of the same qualities that living things have. Viruses are basically just little packets of genetic material. So they're something like RNA or DNA, the same genetic material that we have that keeps us living and breathing and creates all of the information that makes our bodies. But the RNA and DNA inside a virus is just a small snippet that has kind of gone wild. All the snippet says is that it wants to replicate itself over and over and over and over again. So there's not necessarily any malicious content in there. It's not saying, we want to replicate ourselves over and over and over and also kill everything. Basically, it's just saying that it wants to reproduce. And the way that it does that is by infecting cells. And when it runs out of cells in one body, it moves on to the next body and on to the next body and on to the next body until eventually it's spread through a whole population. So viruses are super interesting. And like I said, there's a lot of great resources about them. So I encourage you to look into them, but they aren't alive. So for our purposes, we aren't going to be talking about them very much today, just kind of touching on them there. But what we will be talking about is going to be microscopic life that actually is alive. And so to do that first, we're actually going to look at a data set that is for something that you guys are probably a little more familiar with. So we'll tap the green arrow again, and then we're going to scroll down to the land category and scroll down until we get to vegetation seasonal changes, April 2012 through April 2013. So this data set is really fun. I like it a lot. It's pretty cool. And this data set is just showing basically how plants change over the course of a year. And it's basic information that you're probably familiar with, the fact that plants die in the fall and winter and they grow in the spring and summer. You learned that probably at a pretty early age. But so this data set is showing, like I said, all these different plants. We can actually speed it up a little bit to see the changes over the course of the year. Yeah, you can see in North America there, all the plants are dying in the winter and then coming back in the spring and the summer. But the reason that I'm pulling this up is because of what plants do for us. So everybody probably knows plants go through what's called photosynthesis. So they use the energy from the sun, they take in carbon dioxide, and they end up releasing oxygen as part of the process to make food. We obviously really like that as humans because we breathe oxygen. So photosynthesis is really, really useful to us. But the crazy thing about this data set, all of these plants that we're looking at all over the earth, they're all going through photosynthesis, they're all releasing oxygen, but all of the oxygen that they release from these trees and grasses and flowers and bushes and every type of plant that we're usually used to seeing only ends up producing about 20% of the oxygen that we breathe. About 20% of the oxygen in the atmosphere comes from these land plants. So if only 20% comes from these, where does the other 80% come from? If you were listening at the beginning of the program, you might know where I'm going with this, but it comes from microorganisms, which is pretty crazy. Usually we thank all these trees and land plants and say, oh, thank you for the oxygen, trees, but most of it doesn't come from trees at all. It comes from something completely different. If it doesn't come from land plants, it must come from plants that live somewhere else. So we can tap the green arrow again to go to a new category. That's going to be the water category, which is where all these little microorganisms are living. And then we'll scroll down through here until we get to the phytoplankton model. And so you can see in this data set that there are phytoplankton all over the earth. And this is where that 80% of oxygen comes from, are from phytoplankton just like these. This model is looking at 4 different types of phytoplankton, and so we'll be looking through kind of each one individually. The green and the blue represent just some tiny phytoplankton that are pretty small, but they are 2 of the most dominant phytoplankton on Earth. And when I say really small, I mean very small. They're microscopic, obviously. We're talking about microscopic organisms. The yellow represents flagellates, which are big phytoplankton. As far as you could say big, they're still microscopic. You can only see them underneath a microscope. But they're called flagellates because they have a part of their body that's called flagella, which is basically like a tail that they use to move around. And the red represents a different type of phytoplankton that are called diatoms. Diatoms are really cool. They're some of the largest phytoplankton, and they actually generate most of the oxygen that we have on Earth. They generate about 20 to 50% of the oxygen. So like we said, 20% comes from land plants, 20 to 50% comes from diatoms, and so the rest comes from kind of the other big 3 and other types of phytoplankton. Phytoplankton are called phytoplankton because they photosynthesize. That word phyto at the beginning comes from the same type of word that photosynthesis comes from. They use the sun to make their food. The phytoplankton are also just really important, not only for creating oxygen, but also for creating kind of a baseline of the food chain in the ocean. So the same way that a plant on land might be the base of a food chain here, that an animal eats a plant and then another animal eats that first animal, and on and on and on. Phytoplankton are going to be eaten by something called zooplankton, which are tiny microscopic animals that eat microscopic plants. And then the zooplankton are going to be eaten by something like a jellyfish or a filter feeder, and then on and on from there throughout the whole food chain of the ocean. So the phytoplankton are really, really important for both creating oxygen for us to breathe and also for creating the base of the food chain that provides us with all sorts of fish and provides the ocean with life. Diatoms are important for a whole other reason, though, which is pretty interesting. So diatoms have exoskeletons, which is basically just a skeleton that's on the outside of their body, and it's made out of silica, which is basically glass. So if you see microscopic pictures of these diatoms, they're really, really beautiful. They basically build these little greenhouses for themselves that they live in that have all sorts of cool spikes and different features. But when the diatoms die, the skeletons that they've created still stick around, and they're full of all sorts of different nutrients that the diatom got throughout its life. So when the diatom dies, the skeleton's going to sink down to the bottom of the lake or the ocean, and it's actually able to nourish a whole bunch of new life because it has all sorts of those nutrients inside of it. And so, as a quick kind of aside here, just to talk about how cool diatoms are, we can actually look at another data set that at first glance seems completely unrelated, but is entirely dependent on diatoms. So we can go to the air category, and then we can scroll to the atmospheric chemistry data set, GEOS5 model. And so this data set shows the chemistry of the air in the atmosphere. And at first you might think, yeah, this has nothing to do with microorganisms. You can see that the blue represents sea salt, the yellow represents sulfate, the green represents black organic carbon, which is basically just smoke, and the red represents dust. And so the idea of this data set is tracking all of the different stuff that's in the atmosphere and how it moves throughout the world. But the really cool thing about this data set is if you look specifically at the red, you can see that's supposed to represent dust. And most of the dust in the atmosphere is coming from the Sahara. So it's the Sahara Desert in Africa. It gets picked up by the atmospheric winds, gets pushed all the way across the Atlantic Ocean, and ends up over the Amazon basin. And this dust ends up kind of creating nutrients for the Amazon basin. It's part of the reason that the Amazon is so lush and so green and so full of life, is because this dust goes from the Sahara, travels across the ocean, and lands in the Amazon. But the reason that the dust is so full of nutrients is because of diatoms. So a long, long time ago, where the Sahara Desert is now, there used to be a lake that was called Mega Lake Chad. And like most oceans and lakes and most bodies of waters on Earth, it was full of diatoms. When the lake dried up, the diatoms were dying. They fell to the bottom of the lake where they died fully, but they still had all of their nutrients inside them. So even though the lake dried up, the Sahara Desert is still full of all of these nutritious little diatom skeletons. And so now anytime the wind blows, it picks up the dust in the Sahara, pushes it across the Atlantic, and deposits all those little diatom skeletons, all those little nuggets of nutrients, all over the Amazon basin, and creates the Amazon rainforest. So even the diatoms that are dead are still creating all sorts of cool, interesting life on Earth, which I think is pretty neat. And so we've talked about some of the microscopic life in the ocean, some of the microscopic life that ends up on land and ends up nourishing a lot of our land. But that can seem a little bit far-reaching. It can seem kind of out there a little bit. So for the activity, for a microscope safari, we're going to be bringing microscopic life right to your own backyard. It's going to be an activity where you can look for microscopic life in your own backyard. And so this activity is really fun and requires just a couple of materials that you're going to need at home. But the idea of this is to look through your own backyard for microscopic life. Specifically, I wanted to look for something called tardigrades, which we'll look at pictures of in a little bit. But basically, they're also called water bears or moss piglets. They're really cute, and they're also just really cool microscopic life that is living in your own backyard right now. So I thought it'd be fun to kind of look through for the microscopic life that lives there. So the different materials that you're going to need for your microscope safari, it's all stuff that you should have at home, and there's a few materials that you might need to order online or you could get from a store, but if you don't have them, you don't really need them. So the main materials that you're going to need are going to be some sort of small plastic container. You're going to need at least 2 of these, and preferably they should be stuff that you aren't going to be eating food out of, because you will be squeezing dirt and moss and all sorts of different stuff into them. You're also going to need some sort of long, flat prying tool. You can use a butter knife, a cheese knife, a flathead screwdriver. I ended up just having a long, flat piece of plastic I was able to use, but anything like that should work. You're going to need some water. Doesn't matter what it is, just any water coming from your sink or rainwater or distilled water, doesn't really matter. You're going to need a turkey baster or a pipette or an eyedropper. Again, preferably if you use an eyedropper, it shouldn't be something that's going to end up going into your eye later, because there will be dirt and microorganisms in this. And the last thing you're going to need is the most important. It's a microscope. You might already have a microscope at home. If you do, that's awesome. You can use whatever you have, whether it's a microscope that connects to your computer via USB, if it's a traditional light microscope, it can be anything, just so long as you're able to magnify stuff large enough to see it. There's also lots of great resources to get microscopes online. You can, of course, get them from someplace like Amazon, but there's also lots of great science supply stores you can get microscopes from for pretty reasonably priced. And of course, you can use the microscope for all sorts of different things after this. So those are your required materials. You're definitely going to need those things to do this activity. Other materials that you might not have, but if you do have, could make the activity just a little bit better. Some sort of mesh strainer would be really useful, kind of the same thing that you would put maybe in a kitchen sink to catch food debris. Also, if you have some tissues, you can also use microscope slides, petri dish, or clear nail polish. And those are basically just to make it feel a little bit more sciencey. You can use the petri dish to hold some of your materials and put them underneath the microscope. And then the microscope slides, if you have any blank ones, you could maybe create microscope slides for the things you end up finding on your microscope safari, and be able to look at them later. That's what the clear nail polish is used for too, to help create those microscope slides. But again, if you don't have those things, that's okay. The only things you really need are going to be the 5 materials I listed at the beginning. So to go through this activity, it's going to be pretty simple. Basically, we're going to be using moss as kind of our safari location. We're going on a fantastic safari to the backyard to gather up moss. I ended up having a bunch of moss growing in my backyard. I didn't actually expect that. I was thinking, oh, I'll just go back there and say, oh, you can go on a backyard safari, and then get my moss from someplace else. But there was a bunch of moss growing in my backyard. So you can see the pictures on the left and the right are both different types of moss that I found. Another great place to find moss if you don't have a backyard is going to be sidewalk cracks. So I walked down the sidewalk in front of my house, and I found moss in pretty much every single sidewalk crack that I walked past. So I gathered up some from my backyard, some from the sidewalk. The more moss you gather, the better luck you're going to have with this activity. So after you gather your moss, you're going to hydrate it. And this is where the water comes in. So I put my moss, I pried it up using my prying tool. I ended up using just that flat piece of plastic. Pried it up, and then I put it in one of the plastic containers. If you don't have the mesh strainer or the tissues, all you have to do is just plop it in your container and then add some water on top of it. And the idea of adding the water is that it's going to activate all the little microorganisms that are living in the moss. So things that are living in moss are usually really, really resilient. They're able to dry up when the moss dries up, and then when it rains, they thrive. They're able to reproduce and do all sorts of cool microorganism things. And then when the water goes away, they're able to dry up again, and then they come alive again when the water comes back. So by adding water to your moss, you're going to be just creating a really perfect environment for these microorganisms to live. Now, if you do have a strainer and tissues, at this step, you can put the tissues inside the strainer to create sort of a filter, and then put your moss on top of the tissues. and then put the strainer over the top of your plastic container and just dump your water over the top of the moss in the same way. The difference is that the moss is going to stay inside the strainer and all of the water is going to flush through. But the secret is the water contains all the microorganisms. The only thing you want to keep in mind is that you want the top of your water to be in contact with the bottom of the moss. That way the microorganisms can kind of swim down to the water. They're going to want to live where the most water is because they love water. So you just want to make sure that that connection is there. Again, if you don't have the strainer and the tissues, you don't really need them. It can just make it a little bit easier to find the microorganisms a little bit later on in the process. So after you add your water, whether you're straining it or not, the next step is going to be to wait. And this is the hardest part because you get to watch the moss, but you don't actually get to do anything with it yet. You can wait anywhere between 3 and 24 hours. The longer you wait, the better it's going to be within reason. So I recommend waiting the 24 hours if you can. If you wait too long, the microscopic life might start to die off because it's not getting the right nutrients and things that it needs. So I wouldn't recommend waiting any longer than 24 hours, but waiting about a day is the perfect amount of time for this. It also just lets the moss get all nice and hydrated again. And then we're going to move on to step 4. So after you wait for your 24 hours, you've had this whole long process of gathering your moss and waiting. You're next going to prep your moss. And basically when I say prep, all you're going to end up doing is just squeezing the water out of your moss. And the idea here is that if you have any microorganisms in your moss, you want to try and get all of them out that you can by squeezing all of that water out. If you didn't use the strainer, if you just have your moss in a plastic container, all you have to do is just squeeze the moss water out into that same container and then put your moss someplace else. You can put it in another container and put more water in it and do it again, or you can put it back outside. It doesn't really matter. If you did use the strainer, you're just going to want to squeeze your moss over the top of the strainer and again, strain the water through it a little bit. And then same thing, you can do whatever you'd like to with your moss. And so finally, now that we've gotten all of our water into a container, it's going to be time to start exploring. So all the microorganisms in the water are going to probably sink down to the bottom. So this is where your pipette or your turkey baster or your eyedropper comes in, because you're going to have not a lot of water at this point, but a lot of water to look through with a microscope. So you're going to want to use the turkey baster or pipette, go all the way down into the bottom of your water and just squeeze out a little bit at a time, and then put it into a second container. This is where a petri dish is nice too. I ended up using a petri dish. You can see in my picture there. I used a petri dish, squeezed out the water from the bottom of the container, put it in, and then you're able to look through the water a little bit easier because there's not as much stuff floating around in there. And this is the really fun part. This is when you get to explore, look for all sorts of different microscopic life. You're going to find all sorts of different stuff. The first thing you're probably going to find, though, are things called macroinvertebrates. And so macroinvertebrates are things that you're actually able to see without a microscope. The reason for that is because a lot of macroinvertebrates don't like water. They like it to a certain extent, but they're usually not able to breathe when there's water around. So you might find things like earthworms, for instance. I found some earthworms coming out of my moss because I dunked them in the water and they didn't appreciate that. So the earthworms came to the surface to breathe. I ended up taking them out and actually put them in my eco bottle from a couple of weeks ago. So that was pretty fun. Other things you might end up finding are things like ants, for instance. You might find some sort of small beetles. You're probably going to find a lot of other small insects. If you're worried about having ants and insects and things in your house, I recommend either doing this project outside, or you can just put a lid over your container and they won't escape through the lid. But once they do come out of the moss, you can take those insects and other animals outside, or again, put them in your eco bottle if you're interested. But after you sort out sort of the macroinvertebrates, the big insects and things that you're going to be finding, once you look underneath the microscope, you're going to start finding all sorts of different life. One thing you're definitely going to see for sure, and these are all pictures that I took from my microscopic safari, you're definitely going to see some nematodes. So nematodes are teeny tiny microscopic little worm type things. They're just little microorganisms that live all over the earth. They're in water, they're in soil, they're pretty much everywhere and anywhere that they possibly could be. Nematodes are super interesting. They're used for a lot of scientific research, which is pretty cool because they have pretty simple genomes that we're able to edit. But you can see that the nematodes are moving around, they wiggle around, do all sorts of different cool kind of fun stuff. They're really fun to watch. Um, they're not actually worms. They're the same shape as a worm, but they're not closely related to them. They're their own little microscopic animals. Other types of animals or other types of organisms that you're probably going to see are going to be plants of some kind. So for instance, you might get a little bit of moss squeezed into your water. So moss leaves are actually super fun to look at underneath the microscope. One really cool thing about moss leaves is, like I was talking about earlier with the water, moss is able to kind of dry up when there isn't water around, and then it's able to get nice and plump again when there is water around. And the reason for that is because of these leaves. So each moss leaf is actually just one cell wide. Um, so if you're looking at these leaves, you might think, oh, it's made up of all sorts of different stuff. They're actually just a sheet of cells, basically. And because of this, it means that the water is able to get into the cells super easily, which means that they can dry up or they can expand really easily with water. And when there isn't water around, the cells can lose water really easily, which is how the moss is able to survive the way it does. Another thing you're probably going to see is going to be some sort of algae inside your moss. A lot of algae will live inside moss because it's a nice environment for it. It's a little bit moist, gets a lot of sunlight. So you'll see all sorts of these cool structures. You can see that the one on the right-hand side is kind of pointy. It's got some really cool structures on the inside. The other one's a little bit longer with some rectangles. It's really fun. And you're definitely going to want to look closely at any algae that you see as well, because there might be life living on it. Same thing with the moss leaves as well. I almost scrolled past the algae in the middle. I saw it and then I said, oh, that's nice, and kept my microscope moving. But then I noticed there's little life that's attached to the algae. So you can see where the red text just popped up. It says Vorticella. Vorticella are little organisms that are filter feeders, and they're basically bell-shaped with a little thread on the end that attaches to things like algae or leaves or all sorts of different stuff. Sometimes all the thread ends will attach to each other, and you'll have just like a big ball of Vorticella, which is pretty fun. And they're able to move themselves through the water. So if you find one of those, you're really lucky. That's really cool. But yeah, you can have Vorticella, you can have all sorts of different stuff on your algae, on your moss leaves. So make sure to look really, really closely at the stuff underneath the microscope. And of course, if you're very lucky, you might find some really, really cool life. Everything we've talked about so far is cool, but some of this stuff is just really cool and charismatic looking. So you might find something like rotifers, for instance. Rotifers are over on the left-hand side. Rotifers are super, super cool. I've seen these under the microscope before. I didn't see any this time, unfortunately. They're called rotifers because you can see, at least in the bottom image, it looks like the tops of their bodies are kind of rotating. When these were first discovered underneath the microscope, they were also called wheel animalcules, because the first microscope scientists that were looking at them thought they had little wheels on top of their body. They're not actually wheels. They're basically just little hair-like structures that they use to pump water into their bodies. And along with the water comes all sorts of food for them to eat. So you can see up in the top image too that all those little microorganisms are getting sucked into the rotifer. And then they have jaws that you can see in the bottom GIF that kind of open and close really, really quickly to chomp up all the food that they have bringing in. So rotifers are really cool if you get a chance to see those. Rotifers attach themselves to different things, kind of the same way Vorticella do. So if you're looking for rotifers, you'll want to just wait for a second because they might have kind of closed in on themselves because water was moving around or if you breathed on the water too hard. So you just want to pause just for a couple minutes if you think there might be a rotifer there, and you can see it kind of expand and it'll start bringing in all the food the way that it's doing in these GIFs, which is really fun to watch. And of course, if you're incredibly lucky, you might see something like a tardigrade. Tardigrades are really, really cool. You might have heard about tardigrades before because there's been a lot of different studies on them about what they can survive. Most of these animals, these microorganisms, are able to survive, dry, being wet, the same way that the moss is. But tardigrades are able to survive on a whole other level. They can survive really extreme heat. They can survive really extreme cold. They've even been tested in the vacuum of space. They're able to survive out there too. Tardigrades are just really, really fantastic, really cool, really cute animals. So if you do get a chance to see tardigrades, then you are pretty lucky. But yeah, I hope that you guys have fun on your microscope safari. Yeah. Thank you guys so much for tuning in to the micro program today. We want to thank the MSU FCU Credit Union, the Michigan State Federal Credit Union, for sponsoring the Science on a Sphere exhibit. They, of course, were instrumental in getting the exhibit to the museum. But alongside them, of course, were private donations from people all across the community that really helped us get this exhibit going. I just want to take this time to remind everyone that if you're enjoying these programs, they are provided free for everyone, for teachers, for students, for anybody in our community. But of course, they are not free to make. We do have a link that I'm going to share in the comments of this video. If you guys are able to donate anything to help the museum in general, along with these programs, we really, really appreciate it. We really appreciate your support. But of course, we also just appreciate you tuning in and learning a little bit more about science on a sphere. So thank you so much, and I hope you guys have a wonderful rest of your day.